Hello, I think you can see me from here. I'm Paula. And for today's ProLab lecture, we are pleased to introduce Dr. Olivier Vincent, a physicist at the Institute of Light and Matter in Lyon. Before holding his current position, Olivier obtained his PhD in physics in the University of Grenoble, then worked for 5 years as a research associate in the University of Cornell. His research is very relevant to our group, revolving around transport and phase change in fluid media, cavitation and bubble dynamics, the physics of plants, among other things. Today's lecture is entitled Water in Solutions in Nanoporous Media, Capillarity, Osmosis, and Phase Change. And Olivier, if you're ready, the floor is yours.
Thanks, and thanks, Paula, for the introduction and for the invitation. I think it's a great community to talk to, and it'd be interesting to have feedback on what I will present. So today, what I wanted to present is a combination of different experiments that I've done over the past 5 to 10 years that show some interesting aspects of water and solutions in nanoporous media, both in terms of the dynamics and the thermodynamics. Actually, as you will see, most of this talk can be kind of summarized by playing around with Kelvin equation. And so, of course, for this community, there's not much need to introduce why studying porous media is important. In my experience with porous media, I started to be interested actually from plants. And plants in general have a lot of issues where they need to interact with water. And one of the driving forces for how water interacts with porous media is humidity. And a lot of what I'm going to show today is how porous media interact with humidity, either with fixed humidity or with humidity cycles. And a very important phenomenon that arises in plants is for the transport of water in trees. This is driven by a deficit of humidity in the atmosphere that will generate a driving force from the leaf that will pull up the water column from the roots. This is the phenomenon of evapotranspiration, and here humidity in the atmosphere plays a huge role, where it will drive flow during the day and stop driving flow at night. So that's humidity driving flow through a porous medium. But humidity changes can also drive deformation and generate stresses. And a very famous example is pine cones that open and close depending on the humidity. And also these examples where spores from ferns can actually move and deform under humidity cycles and displace due to this change of shape due to humidity cycles. And there are many other contexts where the interaction of humidity and surfaces or porous media is important. I can cite the atmosphere with aerosols, for example, or the problems of salt solutions that get damaged in porous media in civil engineering, geophysics, or artwork. So a common theme to all these contexts is that the interaction of the water in the porous medium with external humidity generates stresses. And one of the reasons that confined liquids that dry or condense drive stresses is through capillary forces. So of course, if we have a medium that is filled with water with menisci that are flat at the surface, there is not much stress. But if you start drying the system, then you generate curved interfaces, and through Laplace law, this generates a stress in the liquid that can be transmitted to the structure as well and can also drive flow. Of course, the maximum stress you can get depends on the size of the pores, and the smaller the pore size, the larger the stress. And if you get to nanometer size pores, the stresses can be massive, of order 1,000 atmospheres. An interesting aspect as well of these curved confined menisci is that they have a vapor pressure that is different from bulk liquid water. And this is driven, this is explained by the Kelvin equation that relates the vapor pressure or the relative humidity to the capillary pressure in the liquid. And of course, if you have macroscopic menisci, this effect doesn't matter much. But if you go to nanometer nanometric size menisci, then this effect can play a large role, and the vapor pressure deficit that you can get is important. Another way of looking at Kelvin equation is that if you impose humidity over a nanoporous medium, if you fix the humidity, and then you will fix the stress in the medium, and you will achieve some stresses or negative pressure in the liquid or capillary stress in the liquid just by playing on the relative humidity. And the way this gets achieved is because the curvature of these interfaces will reach some equilibrium with the outside humidity, and the lower the humidity, the higher the curvature of this menisci. And this is all described by Kelvin equation, and the stresses that are implied are again very large. Typical examples here is if you are at 100% relative humidity, like here, you have no stress in the liquid. If you go to 90%, you already have minus 145 atmospheres of stress, of negative pressure. And if you go to 50% relative humidity, then you reach 1,000 atmospheres typically. Of course, these stresses can be achieved only if the porous medium has pore small enough to sustain these stresses. And that's why they're important for nanoporous medium. As I said before, these stresses are important, and they can generate mechanical deformations, either in pine cones, but also in solid porous structures like porous silicon here. And you can generate these reversible deformations of the medium just by changing the humidity. And so the humidity is kind of a dial of the stress and strain in this medium. Of course, the stresses can be so large that they can drive boiling, spontaneous boiling on the liquid, which is called cavitation in this case, because the pressure gets much lower, and in fact, much lower than the vapor pressure of the liquid, but in fact, absolutely negative. And this is a highly metastable state, and this can lead to spontaneous bubble nucleation. And this is a movie I actually filmed during my PhD thesis some time ago of one of these cavitation events in a liquid that is at minus 200 bars of negative pressure. And of course, as I said, it's important, these capillary forces interacting with humidity for driving flows in trees. And these capillary flows, because the capillary forces are so large, they can drive large flows, and that are tunable with the humidity. So we will see a bit more later in the talk about these two aspects, cavitation and capillary flows. And I'm going to also look today into what happens when we don't have a pure liquid in the system, but a solution, and typically a salt solution. And this relates to other areas of science and technology, but I would say one of the most prevalent thing where the problem of salty water in porous media is important is heritage conservation and geophysics, where if you have humidity cycles on a porous medium that contains a salt solution, you can drive crystallization or deliquescence of the salts, and this can generate damage and also different problems like salination of soils and etc. And so the solute itself on the liquid has additional effects in compared to Kelvin equation and capillary forces. First, the solute will change the surface tension of the liquid. It will also introduce colligative effects like an effect of osmotic pressure, a change of vapor pressure, etc. And also, of course, it can crystallize or introduce deliquescence in the system, so you can get all these kind of new phenomena that appear. So here, today, I will talk about these different aspects, focusing first on the effect of capillary forces. We will look at evaporation or imbibition, so reverse processes, and the phenomenon of cavitation in some of these systems. And in the second part, I will look at the effect of solutes, looking at the dynamics that induce the solutes, like osmotic flows interacting with the humidity, sorption isotherms that look at what happens in the thermodynamics of the system when you introduce salt, and also the phenomena of crystallization deliquescence. So first, about capillary forces. I'm going to show some experiments that we did mostly with porous silicon, and these experiments were done at Cornell, where I was still there. What we use is porous silicon with that is kind of it's a porous medium that is nanoporous. The pores are typically 3 to 4 nanometers in diameter, but it's also highly isotropic and interconnected. And we have a thin layer of this system where we sandwich this layer between glass and silicon. meaning it's a wafer. Basically, it's a quasi 2D system, and it's open just on one side here. And that's the side where the liquid can evaporate or condense in the system. This can be seen kind of an artificial tree where you have a root system with a reservoir here where the water can be sucked from. You have a porous medium that is kind of similar to the trunk or leaf of a tree system. And here, the evaporating surface is kind of similar to the leaf. And so what we did in terms of experiments with this system is very simple. We fill the system with water. We have the reservoir here. And then we let it dry or let it sit in an environment with controlled humidity. And again, the only way the water from the reservoir here can evaporate or can leave the system is by evaporation from this surface. And we measure the speed of the emptying of the reservoir to measure the flow rate in the system, basically. And the reservoir is not like sketched here. Of course, it's a serpentine channel, and we will see some of its length here. And each line that I show here is an experiment with a different humidity. So when we put the system in a given relative humidity, you see the system starting to empty. So the bright part here is where the gas, and here the liquid. And it's just the reservoir that we see here again. And the emptying of the reservoir informs us that the system is driving flow at different speeds depending on the humidity. I will play again. So you can see, of course, that the lower the humidity, the higher the flow rate. But at low humidity, it starts to saturate, it seems. So if you look at the data of the speed of the flow versus the humidity, you have this first very linear relationship. When you lower the humidity, the flow increases, and then it saturates. And I will look later at the saturation. But the explanation to why the flow is linear is because I didn't plot here the velocity as a function of humidity, but in terms of the driving force that is implied by Kelvin equation, which is the theoretical capillary pressure you get in the pore by equilibrium between the liquid at the edge here and the humidity outside. And so it seems to work very well. So that means that the linear relationship informs us that the Kelvin equation is really driving the flow in the system here. And what it implies as well is that the curvature here, we can tune the curvature of these nanoscale that are at the edge here just by changing the humidity, and the flow responds to that. And the saturation, of course, is, I would say, kind of obvious because you cannot curve the meniscus infinitely. And at some point, you will reach the maximum curvature associated with the size of the pore. And then when you reach that point, the flow doesn't depend on relative humidity anymore because it's just driven by the intrinsic capillary pressure of the pore. So that's the first type of system that we can look at, is drying induced flow in these systems. And of course, what we did is also look at the reverse process of, instead of drying the system, we drive imbibition through it. And the way we do this is we take kind of the exact same system, but without a reservoir. And we just take the system completely dry, and we put it in some relative humidity that is relatively high, I would say. So you see here, for example, 98%, 80%, 50%. And once we do this, so starts from the dry system and increase the relative humidity, you will see that there's a front that develops from the edge in the system. So we are looking at the layer from the top here, and the front dynamics depends on the relative humidity again. And first, it can be surprising that just by raising the humidity, we see a front that invades the system. And the reason that this happens here is because of capillary condensation. And because these nanopores are very small and hydrophilic, there's some critical relative humidity above which the liquid vapor, the water vapor in the atmosphere will spontaneously condense in the pore. And this condensate that you get by capillary forces will start to invade the system. And we did some image analysis to look at the dynamics of this front. And for those who know imbibition, it looks very much like a square root of time behavior that is typical of Lucas-Washburn dynamics. Of course, compared to the standard Lucas-Washburn dynamics, where we have in an idealized single pore we have a meniscus that has some negative capillary pressure that drives flow within the system from a reservoir that's on the outside. Here we have a slightly different system where we have a meniscus from the condensate that's trying to invade the system, but also there's another meniscus at the edge between the liquid and the outside vapor phase that kind of resists the invasion. And of course, the speed at which the meniscus the liquid will invade the system depends a lot on the competition between these two menisci. This meniscus is just driven by the intrinsic capillary pressure of the pore, but again, this meniscus, its curvature is driven by Kelvin equation, and it's fixed by the humidity outside from the equations we saw before. And so the lower the humidity you have, the more curved this meniscus is, and the bigger the competition is with meniscus, and that's why the lower the humidity is, the lower the speed of imbibition, because these two menisci compete more and more. And we could verify that the modified Lucas-Washburn equation that takes into account this Kelvin effect works very well with the data we have for the dynamics of imbibition by capillary condensation at different humidities. And of course, you can even reach a point where the two menisci have the same curvature and there's no invasion. Skip this. So that's capillary invasion with a speed that depends on humidity due to Kelvin equation. And I'm going to look at another phenomenon, looking again at a situation where we dry the system. And here we have a slightly more complex system where we have the same porous medium, nanoporous medium that is sandwiched between silicon and glass, but we also introduce micro cavities or micro channels in the system that are in contact with the nanoporous layer. And we fill the system with water. The water imbibes both the nanoporous layer and the capillary, the water microcavities. And once it's full, we start drying the system. Here we dry the system at 84% relative humidity, which is relatively high, but you will see that still things happen. This is an actual photo of the sample from above, and that's the kind of system we will look at. So here I'm going to show a movie of what happens when we dry this system. And here, probably from your screen, you won't see much, but there's an array of these microcavities, and they're full at the moment, and they become very bright when they empty, and you will see them kind of appearing from the medium. And so when we put the system full at 84% relative humidity, you see that the system starts drying, but it dries by emptying these microcavities that we have in the system. And the interesting aspect here is that if you look at what happens in the nanoporous layer that's below the micro cavities, it remains full at all times, because at these humidities, it will not empty, but these microcavities empty without having a drying front from the edge. And the only way that this can be done is by bubble nucleation or cavitation. And so what happens here is that when you dry the system by the Kelvin effect, you start developing negative pressures at the edge here. And these negative pressures will transmit to the liquid in the pores and then to the microscopic cavities here. And the pressure can be so negative that it drives spontaneous bubble nucleation, and that's how the systems dry. And we could measure that the drying of these cavities, or the cavitation events, happen between -200 to -300 bars of liquid pressure. We also saw that there's some interesting dynamics if you look closely at how these systems empty over time, in the sense that they will have a bunch of cavitation events and then nothing, and then a bunch and then nothing. And there seems to be a periodic emptying of these cavities, separated with periods where nothing happens much. And we could explain this by, I won't go into details here, but this is all due to the nonlinear coupling between the nucleation kinetics, where the rate of nucleation depends on the local pressure, and the pore-elastic evolution of pressure in the system. And this is a simulation of these the dynamics that we simulate in these systems, where each of these red dots is a cavitation events. And then once locally the liquid in the microcavities has emptied, the pressure can go back down again, and you generate these cycles of self-organized bursts of cavitation events that are due to these complex evolution of the pressure field over time, over space and time, actually. So that was a brief overview without too much details of what we did on these systems, of the porous silicon systems, about capillary forces, the flow driven by capillary forces, and the phase change like cavitation induced by capillary forces. And this is several references you can look at if you want to hear more about, read more about these systems. And right now I will move to some more recent studies where we look at the effect of adding salts in these in similar systems. So that's what I'm going to talk about now. And so the first,
just ask a question of the previous.
Yes. Yeah, could I ask you, in the previous one, don't you see any cavitation in the porous silica sublayer?
So we don't see it. It doesn't mean that it doesn't happen, because the scales are so different that there might be tiny bubbles in some locations, but I don't really expect it because of confinement effects. And I expect that the nucleation of the bubbles within these nanopores would take much larger drawing forces to occur. And we can also measure that. So if you look at sorption isotherms of water in these systems, you can see that the layer, the nanoporous layer itself stays full as long as completely full without any little dim due to cavitation, as long as the humidity is above 50 to 60%. And here we dry the system at 84%, so it's way higher than any drying or cavitation in these nanoporous layers.
So essentially that means that the critical radius for a nucleating cavity is too large to develop in the substrate.
Exactly. And so the critical radius depends on the negative pressure you have in the liquid. So it depends on the humidity. And I think the moment at which the critical radius reaches the size of the pores, which is a few nanometers, it should happen around 40% relative humidity. So way lower than, but it's true that it's an effect of comparing the critical nucleus to the pore size, basically.
Okay, thank you.
Sure.
Very nice.
Thanks. So should I move to salts?
Yep.
Let's go. So, this cavitation things is an interesting aspect because it's actually something that happens in trees, and that drives the drying and mortality of trees in the summer, because you have these large drying forces by evaporation in the trees, and you get cavitation events. And one of the questions that was open at that time, and it's still kind of unresolved in the plant science community, is whether once you have cavitation events, you can refill the channels that have emptied by cavitation. And as I said, it's still not resolved whether this happens in plants or not, but we could show that by adding solutes to the water in our systems, we could actually resolve cavitation and refill the microchannels. So here what I'm showing is very similar to the systems I've shown before, except that instead of being these circular cavities, is these long channels, but the system is exactly the same apart from this. The black spots you see here, you can forget about them. It's just some artifacts that are on the external surface of glass, and they don't matter at all in the dynamics here. And so again, when we dry the systems, we have negative pressures developing and we have cavitation, and you will see that each white line that is appearing is a cavitation event that propagates to the channel and then completely empties the channels. And this is the same dynamics as I've shown just before with the circular channels, except with these long channels. And what we did is the liquid that imbibes the systems here is not pure water as before, but it contains a solute. I think in this case it was urea, but it doesn't really matter if it's a urea or salt, and we did it with salt as well. And the interesting thing where when you introduce solutes in the system is that if you, this is driven at, let's say, low relative humidity on the left side, but once you have a solute, you can actually induce condensation within the system by the osmotic effect of the solute, and you can refill these systems. And I will show that when we raise the humidity again after cavitation, we can completely refill these cavities and they completely collapse and the system is full again in the end. And at first, it's not obvious what drives the flow to go within the system to collapse these cavities at first. But if you look, there's some hint at, if you look at the outside surface of these samples here, so these systems, maybe I forgot to say it, but we see it from above, it's closed on the top surface, and the only places where they're exposed to the humidity is the lateral sides here. So here it's open on all four sides, I think. But if you look closely at this side, you see that, I will play the video again. You see that there are little droplets forming on the edge here. And these droplets are actually very important in refilling these systems, and I will show exactly the mechanism just now. And so we could figure out that what happens here is, if you look at the sketch of the system here, it's very simplified. So the big pore on the left here is actually one of these microchannels that I've shown before. The little pore here schematizes the nanoporous layer, and this is the external surface here. And the thing is, when you have a solute in the system, you can impose a vapor pressure that is above the vapor pressure of the solution. If you have pure water, the equilibrium is at 100% relative humidity, and you will have to impose a supersaturated vapor to induce condensation. But here with a solute, the solute lowers the vapor pressure. It's one of these colligative effects I've talked about in the introduction. And due to this, if you have a solute and sufficiently high relative humidity, you can induce vapor condensation in the solution due to this osmotic driving force, or this colligative driving force. And if you do that and you have a hydrophilic system, the condensation will tend to make the solution spread out of the system and actually flow out. And once the solution flows out, it forms either films or droplets on the external surface. And due to the equilibrium between this film of solution or the droplet solution and the humidity, you will tend to dilute the solution, because once again, we are imposing a humidity above the vapor pressure of the solution, and so it will drive condensation into the solution as long as the solution is not dilute enough. And this dilution of the solution on the outside surface makes it less concentrated than the solution that's still in the inside of the system, and this drives a solute concentration gradient that drive an osmotic effect. And if the pores are small enough, this osmotic effect can be quite large. And this is actually what drives the filling of these microchannels after cavitation in this case. And of course, an interesting aspect when you see this is that you can think that what happens if you have the reverse effect. So here we impose a high relative humidity on the outside, but you can say, oh, let's impose a low relative humidity. And of course, if you start with a system that has a meniscus here and impose a low relative humidity, you will drive drying and cavitation. But if you already have a film of solution formed on the outside of the system, what you will drive first is not cavitation, but a concentration of that solute film, solution film. And by concentrating the solution film here more than the inside, you will drive an osmotic effect that is opposite to before. And the osmotic driving force here is from the inside to the outside. So we'll tend to empty the system instead of refilling it. And we check that this happens by, we just played around with a sample here with a sample where we just have one of these cavities that is still empty. And by similar to before, when we impose high relative humidity, it will shrink the bubble. But when we switch back and forth between high and low relative humidity, we can trigger reversible filling and emptying of these cavities just by an osmotic effect. And I won't go into details here, but this is what we measured. And you see that we have a very steady, very quickly, we have a steady state driving flow, osmotically driven flow in these systems that respond very quickly to the humidity changes we impose. Okay, and so that's interesting because we can show that we have first a very high control of the flow rate in the system of filling and emptying just by the humidity, and also that we can refill systems completely by osmosis. And the systems I've shown before up to now where we added salts are systems where we fill and we empty and refill cavities in the system that are macroscopic, but we don't look at the drying of the nanoporous layer itself and what happens when this happens. And of course, to dry these nanoporous systems, we need to impose very low relative humidities, again, because of the very small pore size. And what we did to just evaluate what happens in these systems with the complete range of relative humidities, but within the nanopores themselves, is water absorption isotherms, which is just a measure of the water content in the system at different relative humidities. And we cycle the humidity between very low, let's say 20 to 30%, and to very high humidities close to 100%. And this is well known that if you have a nanoporous medium and you cycle humidity above it, when it's at low relative humidity, the medium is basically dry with potentially some absorbed molecules on the pore walls, but not bulk liquid in it. If you increase the relative humidity by going to the right here, at some point you will get capillary condensation that I talked about before, and that drives capillary imbibition in some cases. And so here is because the medium is hydrophilic with small pores. The condensation of liquid in the system is at relative humidities lower than 100%. Here, the pores are about 10 nanometers, and the condensation is around 90% relative humidity. And once in condensed, you have a reversible plateau here where it stays full no matter what the relative humidity is. And if you go to the left and start drying the system by lowering the relative humidity, there's some hysteresis, but it evaporates also at a humidity that is lower than 100%, and here, which is around 85%. And so this is when the systems contain no salts. So it's just a nanoporous medium that interacts with humidity. And we looked at what happens when we introduce salt, and things change a lot. First, so the curve with salt is this one. And you see that first it's shifted to much lower relative humidities, which is in itself not surprising because, as I said before, when we introduce salt, you lower the vapor pressure of the liquid itself. So all the humidities are kind of shifted to the left. But also you see new shapes that appear in the system. You have condensation here again, but then condensation is not complete, and it continues here until the sample is full. There are some things here that I won't talk about much, but then when you dry the system, instead of drying very quickly like here in terms of the humidity, so this system, there's a very sharp transition between wet and dry. Here, there's a very smooth transition. So the systems empty very smoothly, I would say, and until there's a very sharp kink in the curve where the systems empties abruptly up to its dry state. And we can cycle, and this is reversible. And the reason for all of these effects is because of the solute, of course. And what happens here is that we start from a system that contains a solution, and the solution fills the complete volume of the pores. And here we reach a point where the curvature is too large, and the system starts drying. But of course, when drying the system, we concentrate the solute, and this reaches a new equilibrium where the system is happy in equilibrium with the outside humidity. But just we concentrate, when we lower the humidity, we concentrate the system just more and more. But the concentration cannot increase indefinitely. And at some point, there will be crystallization because the concentration is too high. So the little kink we see here is the abrupt crystallization of the salt within the system. And the interesting thing here is that salt is known that when you take a bulk salt crystal and increase the relative humidity, there's a critical humidity at which it will start absorbing the water around and spontaneously dissolving from the water vapor in the atmosphere. And for NaCl that we used here, it's about 75% relative humidity. And here we see that the uptake of water in the system is much lower than 75% relative humidity, which would be around here. And meaning that there's something that is shifting the deliquescence point in the system, and of course it's confinement. And so the message here is that by introducing solutes, you change a lot the absorption properties of water in these nanoporous layers, and you also see the phase transitions of the solute that are shifted compared to the bulk, and we see clearly crystallization here and deliquescence here. And the interesting aspect is, so I won't go into details in these curves here, but we did cycles with a lot of different concentrations of the solution. And what's obvious is that no matter what's the amount of salt in the system, the total amount of salt or the initial concentration you will use to fill the system, the crystallization point in terms of humidity is always at the same spot, and the deliquescence point is also always at the same spot, which means that no matter the concentration we use, we can define a confined crystallization point and a confined deliquescence point. And we did a systematic study of where these points sit, depending on the characteristics of the porous medium and especially the pore size. And so that's what we did here. We looked at how the deliquescence point in terms of humidity and the crystallization point in terms of humidity depend on the pore size. And we use different types of porous silicon here. That's actually the same porous silicon as I've shown for the previous experiments for the osmotic dynamics and cavitation dynamics. We have some other types of porous silicon here, and we have some porous alumina oxide here, AAO. And so what we see here is that so that's bulk deliquescence here, 75%, as I mentioned before. And you see that when you reach pore sizes that are, let's say, above a few tens of nanometers, you get very close to bulk deliquescence. But once you start going below 20 to 10 nanometers, the deliquescence points is very, very shifted compared to the bulk. And there's the same behavior for crystallization. And again, the reason for this is solely Kelvin equation. And we could fit this curve here to the data points with a modified version of Kelvin equation, but without any adjustable parameter. The only modifications we have to put in Kelvin equation is an activity term that takes into account the osmotic effect of the solute or the colligative chemical potential effect that the solute has on the liquid, and also the fact that the surface tension of the liquid changes because of the solute. And just with this equation, we can fit this data without any adjustable parameter. And the same happens for crystallization, except that here there's an additional fitting parameter. I mean, there's one fitting parameter, which is the supersaturation at which the solute crystallizes in the pore. And the supersaturation is, I mean, for NaCl solutions, the maximum supersaturations that have been measured by levitating water droplets very carefully and drying them slowly is around 2.2, and we reach bulk supersaturations, I mean supersaturations in our system that are very similar to the maximum supersaturations that we can measure very carefully for NaCl in control systems. So the thing is, the interesting thing I would say is, again, by just applying Kelvin equations to these systems, we can very successfully describe what's happening in terms of the phase change of salt in these systems. And this can have interesting consequences for the deliquescence and crystallization damages that we can observe for real materials, for example. And I'm gonna finish this presentation by just a few examples of what we are doing now with similar systems. And actually, we noticed that if we just look at our systems with a camera when the system is around here in terms of the cycles, just before crystallization, we can actually see the crystallization happening in the layer. And this is a video that we took of crystallization appearing in nanoporous silicon, just by looking with the camera and with some contrast enhancement. And so you see that you start to get these nucleation spots in several parts in the system, and these nucleation spots grow then in a circular fashion and then merge. So don't really pay attention to this dark circle in the center. It's an artifact due to some other experiments that I'm going to show you a bit later. But so right now we're looking at these nucleation and growth dynamics and trying to extract the nucleation rate and the growth rate dynamics and look at how confinement. So all these crystals that we see are basically growing in this nanoporous sponge, and we are trying to see whether there's any interesting dynamics in the growth dynamics and in the nucleation pattern that we see here. So that's one of the directions we have right now. Another one, which is actually the reason why there's this dark spot in the center, is that we also look at what happens when we have drops impacting or just deposited on these nanoporous layers, drop of salt solutions, and how they imbibe and dry depending on the humidity and stuff like that. And so here's a movie where you see a droplet of NaCl solution deposited in one of these nanoporous layers, and you see a lot of things appearing, but first you have a circular imbibition around the droplet, and due to the fact that it's also drying at the same time due to evaporation, you have crystallization. And we are trying to look at the patterns we get of crystallization both outside and inside of the system, depending on a lot of parameters. And the last thing I wanted to show you is once we have this salt deposit basically on top of the system, on top of the nanoporous layers, we also look at what the effect of humidity cycles have on these systems. And for example, here is we start from these deposits and we increase the humidity and then we will lower it again. And you see that there's lots of things appearing. There's this spontaneous deliquescence of the salt on the outside. There's also deliquescence inside, and then that forms these little droplets, and the droplets grow and merge. And there's lots of funny dynamics here. And when we dry the system, you see all the droplets disappearing and another deposit that is formed that is very different from the initial deposit. So cycling humidity in these systems that have salt on top or salt droplets raises some interesting questions of how the deposit will evolve as a function of the cycles. Okay, so that's I'm reaching the end of my presentation here. So what I've shown is different experiments, but the common theme is the behavior and dynamics of water and solutions in nanopores. And what I've shown is a lot of its dynamics and thermodynamics has to do with the Kelvin-Laplace equation. And the Kelvin-Laplace equation entails capillary flows, like imbibition or evaporation, like I've shown. It can also drive phase transitions. I've shown examples of cavitation, crystallization, and deliquescence. And also, it can couple to other effects like osmosis or pore-elasticity to generate dynamics and patterns. So finally, I would like to acknowledge the different people that have worked with me on these themes, both at Cornell University and here in Lyon. And I know that very soon there will be a postdoc position available and open for working on these themes. So if you are interested, please contact me. And this is the funding we have right now for developing these themes. So thank you for your attention and be happy to take any questions.